My name is Daniel Madison. I am currently a professor at Stanford School of Medicine. I've been that for about over 30 years. I went to school at UC Irvine for my bachelor's degree. And then I went to UCSF for my PhD. And then I did a postdoctoral fellowship at Yale School of Medicine. And then I came here. I started in research my freshman year as an undergraduate. So that's a long time ago. Really got into it pretty early, kind of almost by chance. When I was applying to colleges, I had applied to UCI. I was sitting at home, when I was in high school, and I was sitting at home, and my mom dropped a newspaper in front of me and said, read this article. And it was about this guy at UC Irvine who was doing this research. And I just thought it was the most amazing thing I'd ever read about. And then by pure serendipity, it turned out I had him for the first class that I took there. And so I basically worked really hard to do really well in that class. And then I hit him up after that to let me work in his lab. And he did. And that was 50 years ago, almost
Wow.
49 years ago. So yeah, so I've been doing it ever since.
What have you enjoyed most about doing research for now 50 years?
I really it's kind of hard to put my finger on the thing I like best. There are lots of things I really like about it. I think the most exciting part of it is that I have been privileged through my work to basically be an explorer. And I know that in the course of my work over the years, and this is not unique to me, this is really true of many, probably in most, decent scientists, that you have a lot of opportunities to see something that nobody has ever seen before in the history of the world. You need, sometimes it's a small thing or sometimes it's a bigger thing, but basically you're looking at things and going, wow, in the whole history of the world, I'm the only one that's ever seen that. To me, that's the most exciting part of it. The other things I really like about it is really just, it's a career where it's very busy in terms of just you're always working, but it really has a lot of flexibility. So, if you can get into a position like I'm in, a faculty member at a college or university, you really have a super large amount of freedom to do whatever you want.
Mm-hmm.
And to me, that's paradise. I mean, I like doing whatever I want. So, my scientific direction is entirely my own.
What has been your biggest challenge? And how did you overcome?
I think the biggest challenge really was getting to the place where I am. Faculty jobs are very hard to get. They're very rare, in terms of, they're rare compared to the number of people. And I really just, through a combination of obviously hard work and making some good scientific findings that interested people, hiring people who would hire me,
Yeah.
obviously overcame that. But a lot of that was luck too. I mean, just kind of being in the right place at the right time. And honestly, the way I always remember when I was just finishing up my bachelor's degree, I'd been doing research for several years at that point. So I kind of started to hang out with the graduate students because we were kind of working on the same stuff. And I remember sitting out in a bar with about 15 of them one night. And the discussion was, which one of us is going to get a faculty job? And I remember thinking about that and going, well, that sounds like a really grim prospect for a career. And then I just thought, well, you know what? It's what I want to do. And I don't really want to do anything else. So I guess I'll just go for it. So that's how I overcame it. I just didn't worry about it. I just went for it. To me, that was probably the key to it. And, it's worked out. So as the famous hockey player Wayne Gretzky said, you miss all the shots you don't take.
Out of all of the research you've been a part of, if you could just explain one of the projects you've worked on.
This is the headquarters of the Stanford School of Medicine, located obviously in Stanford, California, not too far from you. I work in this building right here. This is called the Beckman Center for Molecular and Genetic Medicine. And my particular laboratory is basically right behind this tree. And so that's where I work. These are kind of just a survey of the general areas that have interested me over the years. I'm what's called an electrophysiologist. So I use electrodes to poke into brain cells and basically record their electrical activity and kind of learn how they behave in the face of different stimuli and drugs and that sort of thing. I've been interested in the basic mechanisms of synaptic transmission in the brain, but you know what synaptic transmission is?
Yes, yeah.
So, and I work on something called synaptic plasticity, which is how synapses change in the face of experience. I've worked on that a long time, done some work on the actions of cannabinoids and the Alzheimer's important molecule amyloid beta. And right now we're working on a function of a specific subtype of neuron, an inhibitory interneuron in the cortex of the brain. We're studying studying that because it's related, the function of that neuron is related to a variety of brain functions and disorders, in particular dissociative disorders. So dissociative disorder is basically almost like an out-of-body experience where you don't feel like you're yourself, and we all have them. We all have these weird things that happen, right? Like deja vu and maybe a little feeling that you're outside yourself or something. And then, that's just normal everyday life. And then there's disorders where it's really a problem, where it's that way for you all the time. Feel like you're outside your body, looking at yourself. That's a dissociative disorder, or schizophrenia is a dissociative disorder. These nerve cells in particular really seem to be very important in terms of controlling that behavior. Basically, what I want to do is I want to sort of show you just a couple of techniques we use because they're important for understanding. Anyway, we do electrophysiology, which is, as I said, poking electrodes into things. But we also have added this technique called array tomography. So array tomography is an anatomical technique where you can reconstruct objects inside a piece of tissue digitally. And the way you do it is you take some kind of tissue, in our case, it's brain. We usually work on brain. And you take that tissue and we fix it, which means you put it in a chemical like formaldehyde to preserve it. And then you embed it in plastic. And then you put it on a machine called a microtome, which is sort of schematized here, which is just a knife that goes up and down like this. And it feeds that tissue into the knife. And it makes this string of sections. So it goes over and over and over again. And every time it goes, a new section gets added. And then it just keeps growing and growing until you get what we call an array of sections. So it's just a string of very thin cuts. Almost think of it like you're at the deli and you have a big salami and the guy is slicing it up, right? So that's what we do.
Yeah.
Except in this case, we've treated it so the slices stick to each other edge to edge. The important part of that is that each section in this array is a serial section, right? So they're in order. So basically what we can do is we can take those arrays, we can put them on a piece of glass, we can stain them in a variety of ways, and then we just image each section individually down the line with a specialized microscope, and you end up with a stack of images which are in the right order, right, because of the serial section. And then you can essentially take those images and flatten them down into a single image, and you get this result here, which is just a picture of whatever it is you're looking at inside that tissue. And you can get down to pretty low resolution. I'll show you a better picture of this later, where you can see very small things inside that piece of tissue. The thing that this technique, array tomography, is mostly been used for is really just taking a section of brain and just asking a question like, what's in there? Here's a big section of brain. I want to see what's in there. And this is an example. This is a section of brain which has been stained with three things. So it's been stained with the blue stain, which is a nuclear stain. It stains the nuclei of nerve cells and other cells. This is a specialized dye for a particular molecule that's found in this particular kind of cell right there. And then the white here is myelin. This is the way people normally use it. They want to say, well, what's in there? And are there interesting associations between things they see and whatever? We took a completely different approach. And the approach we took was we didn't want to see everything. We just wanted to see specific things. The way that we approach that is we start out with a piece of brain. So you can see right there that little lit up thing in this chamber right here underneath the microscope. That's a little slice of brain. There's an electrode coming from this side and there's an electrode coming from this side. What we're basically doing is we're poking two neurons in this piece of brain. So with the microscope, we can see them individually. So this right here, this sort of shape right here, that's a nerve cell. And this shape right here, that's another nerve cell. And you can see an electrode coming down here and poking into that one, and an electrode coming down here and poking into that one. And then you might also notice here that the electrode here is pink and the other one here is kind of yellow-green. And that's because we filled those with dye and we're injecting that dye inside those cells. So then when we do the array tomography, that's all we see, right? We see just those two cells. And the nuclear stain, we did the nuclear stain too. That's actually the nuclear stain is used to register the images when you stack them, right? Because you have to be able to line them up. And so those are the profiles we use to line them up. And so instead of, like looking at everything, we just wanted to focus in on like specific things. And then also because we have these electrodes in the two different cells, we can actually look at the physiology between them. So the two cells, this one and this one, actually have a synapse made between them. if we cause one cell to discharge an action potential, then we can see a synaptic response in the other one. That's basically the sort of almost like the quantal circuit in a brain. It's one cell talking to one other cell. And then the idea is we take that, record all the physiology we want, and then we subject it to array tomography and then reconstruct it. And then we're trying to see if we can find things in there that correspond to the physiology that we saw. This is just a picture of the rig that we use. So this is the recording apparatus. I have 5 of these in my lab.
Oh, wow.
So this is the microscope here. These are the 2 electrodes. They don't have electrodes on them right now. They're sort of just waiting for somebody to come and use them. And then everything else here, this is just nutrient solutions that you flow in to keep the brain alive. And anyway, you just do that experiment. And then when you're done, you pull it out, throw it in formaldehyde, and you do array tomography. So this is the kind of image that you can actually get. So here's 2 neurons that we injected with the red and the green dye. The red dye is actually a specialized dye that we buy from a company that sells dyes to biologists. It's called an Alexa dye. They're really amazing there. And then this is one called Lucifer Yellow, which comes from fireflies. Basically part of what makes fireflies glow. And so you can see the 2 cells. And then what we did was we found every place that there was a synapse made between the red cell and the green cell. We can zero in on that. So you can get this kind of really amazing resolution where this is a synapse here. This is the transmitting side of the synapse right here. And this is the receiving side right here. And we can basically go and we can record, the physiology of those, and we can do experiments, and then we can go in and see, is there anything interesting in the anatomy that explains what we saw in the physiology? This is the latest work that I was telling you about. This is the interneuron work. The pink cell here is what's called a parvalbumin interneuron. It's, like I said, they're really specialized neurons that are in the neocortex, the new cortex on, that rests on top of your brain. And those are the ones that really control what's something called gamma rhythm. So you know that the brain has these rhythms, right? Alpha rhythm, beta rhythm, theta rhythm. Yeah. Gamma rhythm is the really fast one. It goes at about 50 to 100 times a second, 50 or 100 hertz. And that's the rhythm that seems to be important for dissociative disorders. When you are maintaining that high frequency, then you're fine. And if that gets disrupted, you're not fine. So we're trying to find out why that is. Basically, what is it about the rate of firing and the communication that leads to that? So, here's a microscope image of 2 nerve cells with their electrodes coming down onto them. This is what the discharge looks like if we cause it to fire. Again, we can do these paired recordings because these cells are synaptically connected. So here's us firing an action potential in one of the cells, and here's the response in the other cell. And so, like I said, and then we can completely reconstruct these cells. So, we can find the synapses. We can do, this is this flattened image of 100 sections. These are individual sections. Individual sections. And so what we do is we find the synapse, and then we go through the individual sections and we stain them with an antibody for particular proteins that are only found at synapses. And so that's what we're showing here. So gefrin is a synaptic protein that's found on the receiving cell, and synapsin is one that's found on the transmitting cell. And as you can see here, we have both of them present there. So we know that's a synapse. And then we can reconstruct the entire structure of that cell. And so this is the axon branching pattern. The blue parts are where it's myelinated. And then this is the target cell. You can see where the synapses from this cell all terminate. So we have a lot of information about how these really sort of quantal neural circuits are organized. And then the last thing we're doing technically is we added another step because this experiment wasn't quite hard enough already. So we have the electrophysiology, we have the array tomography, but before we fix the tissue, we're now also sucking the nucleus out of the cell and processing it for RNA sequencing. And so basically, if we sequence all the mRNAs that the cell is making, then we essentially know what genes are being expressed. And so that's just another piece of information we can use to understand the function of what's going on in these cells. We have the physiology, we have the anatomy, and now we have the genetic gene expression. And so we're trying to synthesize all that now into a complete picture of how dissociative disorders occur. And I think that's all I have. So that's all I prepared.
For like high school students, how would you recommend them getting interested in research?
Yeah, no, I think it's a good thing to obviously do. I don't know that it's really, other than learning yourself what you want to do, I don't think it's really critical to get actual lab experience in high school. As you said, it's really hard to do, and they've made it harder. We used to do it more, but about 5 years ago, they put this whole Minor Protection Act thing into place. And Now, if I want to have an undergraduate working in my lab, I got to jump through some pretty extreme hoops, just to
Yeah.
make sure I'm not molesting anybody. Um, but in any case, it's not bad to get. I mean, obviously, any more the better, but it's not too late to start basically once you get to college,
Mhm.
but it's good to know it's what you want to do, right? So, you probably won't have the same experience I had in terms of, piquing my interest. Um, but if you can do what you want to make sure that you are interested.
Mhm.
But even then, you can always try it as an undergraduate. And if you don't like it, you don't like it. But I think that's a really important thing, actually, is that, there are a lot of people that are interested in science, just don't like lab work. There's only one way to find out if you do or you don't. And there are
Mhm.
other things you can do if you don't. But if you want to be a, wet lab bench scientist, it's not for everybody. It's very hard work and it can be very tedious and it can be very frustrating. And honestly, I think, you asked me what my challenges were. And if I had to add a third one, it's just dealing with all the failed experiments. It's just part of the territory. You just got to deal with it. And honestly, it's 90%, 80 or 90% of experiments fail in one way or another, or they don't give you the results you needed, or they don't give you enough information or whatever. And then every once in a while, you just manage to cobble together some really amazing ones that work. But there's always a lot of failure on the way. And that's probably, I think, what a lot of people find difficult about it is just dealing with all the failure. But you kind of got to think of it like a baseball player, right? So maybe you only hit the ball 2.5 times out of 10, but that's actually pretty good.
In your experience in research, what have you noticed, like in a researcher, like what traits define a great researcher in STEM?
I don't know that there's really a single, thing I could say. Curiosity is important. Ethics are incredibly important. One thing you learn when you do this for a while, it's really easy to fool yourself,
Mm.
especially when you've got a favorite idea. It kind of go either way on an interpretation of a result. You got to be really rigorous about making sure you really demonstrated something, and that kind of just combination of curiosity and dedication to the truth is really critical.
Mhm.
You got to be willing to work hard. I mean, that's a lot of places, but a typical young scientist in an academic setting is probably working 60 or 70 hours a week. And even at my advanced age, I'm still putting in 55 or 60. But, as they say, if you love what you're doing, it's not really work. So, but I want to return to one thing before we finish up. And that's just, if you're interested in a career in research, it's critical that you get into a lab as an undergraduate. You're just not going to get anywhere without that. When we're looking at people for grad school, so, to earn a PhD, which is obviously the next step after the undergraduate, that's the first thing we look at. So we get applicants in and, the grades are generally uniformly pretty good, and test scores, we don't even look at anymore because we were kind of useless. But, letters of recommendation, and you got to get all that stuff. But the first thing we look at is what's the research experience. And if you don't have that, you're just going to go right to the bottom of the pile in terms of, applications and getting into a school. So number one piece of advice is, if this is a career you want to pursue, you can't get into a lab too early as an undergraduate, because, when you come up against it in terms of like getting into grad school or stuff, your competitions can have 3 years of experience or 2 years of experience.
Yeah.
And if you've been, if you're like I did a summer in a lab, we're kind of like, okay, that's nice, but there's this other person who's
Mhm.
did 2 years, right?
Yeah.
And it's important for two reasons. I mean, one is it shows your aptitude, but it also shows your interest, right? I mean, it can be a slog, and not everybody likes working in a lab. And if you've only worked in the lab for a month, you don't know yet whether you like it or not.
Mhm.
So I really highly, highly, highly can't recommend it too much. That you need to get into a lab as soon as you can.
Mhm.
Yeah, maybe you can give the freshman year a rest, but you don't have to. You need to get adjusted to college and make sure you're doing okay in your classes and everything, so you don't want to overburden yourself. But
Yeah.
I would not wait longer than maybe junior year to get into a lab.
Mhm.
If you really want to pursue this as a career, that's really a necessary step. So, and obviously you got to take the classes that
Yeah.
background for whatever you're interested in, right? We look at that too, right? Have they taken any neuroscience classes, right? How do they even know they like this? That would be the advice that I would absolutely offer.
So thank you so much for your time. It was really great talking to you.